Christina.
So good morning, everybody. Again, I'm Christina Conroy, and as Scott said, I'm a final year CTP PhD student. And unusually today, I will be talking about a questionnaire which I've undertaken on SWD. So this is the Understanding Current D. Suzuki Strategies and Identifying Barriers to Adopting Novel Management Plans. So as always, I'd like to thank my project funders, the BBSSC and the AHDB, the CTP for providing additional training and funding opportunities, as well as the internship which I undertook with Berry Gardens. So Drosophila Suzuki is the major European horticultural pest. Now, the reason for this is that it has a highly serrated ovipositor, which can be seen in the bottom right-hand corner of the screen circled. Now, this allows the Drosophila to cut into ripe and ripening fruit, unlike other UK Drosophila, which can only lay their eggs in rotting fruit. And then you get this fruit degradation caused by larval feeding inside the fruit, and then secondary infections brought in via that hole created during oviposition. One of the things that I think makes this project so incredibly interesting, and SWD in general, is the fact that there is a summer and a winter moth variety. And to make this clear, they are a single species, but two moths. So the summer moth transitions into the winter moth and responds to reduced light and reduced temperature. Now, the winter moth is significantly larger, darker, and exhibits delayed ovary development. But from listening to my previous talks, I'm sure you're all aware that the winter and summer moth often respond to chemicals in different behavioral ways. So my question here is really, how does this affect management plans in both the short and long term? Moving on to the actual topic of my conversation today. So why a questionnaire? A lot of questionnaires which have been undertaken have been incredibly narrow in the thing that they're actually researching. So there's been a lot of barriers of adoption questionnaires. However, these have all been undertaken in sub-Saharan Africa and therefore not necessarily relevant to the UK industry. And similarly, the questionnaires which have been undertaken on SWD in the UK have been very narrow in the focus, as previously mentioned. So I undertook a CAP questionnaire. So this is knowledge, attitudes, and practices on D. Suzuki and IPM. So the idea here was to work with the industry to, first of all, understand the barriers to adopting new strategies, to reduce the time between design and implementation of said strategy, with the overall aim of increasing product affordability, which would obviously lead to a far more marketable product in the long term. So as part of this, obviously I collected a plethora of data, but due to the time constraints which I've got today, I'm just going to be going through three of the shorter objectives. So first of all, to identify implemented IPM strategies, then to determine participant satisfaction with those available strategies, and then identifying the barriers of adopting these new strategies. The methodology for this was quite straightforward. First of all, I had to seek ethical approval, and this was made sure that it was in line with governmental guidelines, as well as the University Research Ethics Committee. Then a questionnaire was created, and this was undertaken on 3 growers and 2 academics. And then all the information from that pilot study was then fed back into the final questionnaire, and with the help of Scott Raffel, was fed to 7 different grower organizations. Moving on to objective 1. So the implemented IPM strategies. The most frequently implemented IPM strategies within the respondents of my questionnaire was, first of all, good hygiene used on SWD, plant protection products, and then traps used for both monitoring and precision monitoring. So with the latter, which trap large numbers of SWD. And then the most significant cost to SWD was, first of all, labor, and secondly, plant protection products. And this had over 60% of the participants spending over £2,500 per hectare per annum. Then moving on to the satisfaction with the IPM strategies. So overall, there was an interest in the adoption of either new or alternative strategies which are already available. To state the obvious, the people who were most interested in a new strategy were the people who had SWD, which was a problem on their farm. But what I found was really interesting was there was a winter moth effect. Now, this was basically when the participants were specifically aware of the winter moth and the biology and other surrounding information on them. The people who were aware of this information were far more likely to be interested in adopting a strategy which focused not only on the summer moth, but on the winter moth as well. And this wasn't just for monitoring purposes, this was controlling the winter moth to better control the summer. But overall, everybody who was a part of this questionnaire was interested in new control strategies for SWD. And I don't think that's a particular surprise to anybody, with 70.4% interested, as I was speaking about, this year-round strategy that's not reactive, but instead proactive. And then onto the barriers of adopting new strategies. So people were far more likely to adopt an IPM strategy if they understood the technology better, and very unsurprisingly, if the IPM strategy used fewer resources or money. Now, resources here, I may also be speaking about labor, machinery, and things like that, which don't necessarily have an individual monetary amount. However, interestingly, more resources were deemed acceptable, or more money was deemed acceptable to be used if the participant understood the science behind the strategy and if they had positive social interactions with other growers. And what I mean by a positive interaction is, for example, when you speak to another grower and you say, oh, I had an incredible time when using, this particular product. Or the converse is also true, oh, this product wasn't effective at all. That would be considered a negative interaction. And then the people who were less likely to adopt an IPM strategy were those who had these negative interactions with their colleagues, if the strategy cost significantly more, and if there was more resources required. In terms of adopting a completely novel strategy, from the questionnaire, it became very obvious that this was going to be sort of a three-step process. So first of all, a new and novel strategy would need to be established on a small number of farms. And these people, these pioneers, would then need to be offered multiple training opportunities. So this could be in terms of speaking to the agronomists, speaking to scientists, attending events such as this, or online information. From here, in order for it to go to a second wave and for it to be established on a larger number of farms, grower-led training would be the way forward. This is what most participants were actually interested in hearing from their colleagues. And as well as this, at the same time, we need to be really aware about the infrastructural needs of individual farms. So obviously there are small farms, there are large farms, there are farms which are in different environments. And I think that this is something that everybody was really concerned about, that we needed to get this right. So that would be occurring in the second stage of this process. And then finally, in order to get into a country-wide establishment, we would need to have additional access to scientific advisors. So a lot of the research said that if they understood how the strategy works better, they were far more likely to implement the strategy, with additional emphasis on how the strategy actually works. So this could be the science behind the strategy or how it works on their farm as an individual. So in conclusion, as I was saying, this is really a small snapshot of the information which I gathered. I could have spoken about this for the better part of an hour and still only covered half of the information. But moving forward, I think that we really need to be undertaking cost-benefit analysis or techno-economic analysis in order to really flesh out potential novel research plans so that growers have more of an idea of how this is going to financially affect them. And then quite excitingly, this three-step plan, whilst I've sort of done this in regards to SWD, this could aid the application of any relevant control technology within the horticultural industry. So thank you very much for listening to me, and I apologize about the problems that we had at the start there. but for my supervisors at NRI and NIAB EMR, so Dr. Daniel Bray, Professor David Hall, Dr. Charles Whitfield, and Dr. Michelle Fountain, with special thanks to Scott Raffle and Herrick Duncalf, who has made everything that I've done within my PhD possible. So thank you very much. Christina. If you have any questions.
Thank you very much indeed. There don't appear to be any questions at the moment, but one point I would make is part of my job, and one thing that we've been trying to do in recent times is to disseminate the results through agronomists to help to train growers. And I think that system is in place, and I think there is potential to do more of that in future. So I would hope that will work seamlessly. Just as a reminder to everyone, if you have got any questions, please do post them in the chat room. We are running slightly over already, so Christina, thank you so much. Christina, we bid you farewell because we've seen you on our screens many times in recent years. I should just say to everyone, Christina has now finished her PhD. I think you're writing up now, or just about, or have you written up?
I'm about to hand in my thesis in the next 2 or 3 weeks.
So wherever life leads you in the future, please keep in touch, and we hope to see you at some point in the research world of SWD or related subjects in future. Thank you so much, Christina.
Thank you very much.
So we'll move swiftly on. Our second speaker is Adam Walker. Again, Adam is known to most of you from the work that he's been doing in the NIAB EMR entomology team. He's been working for the last couple of years on the opportunity, shall we call it, to reduce overwintering populations of spotted winged Drosophila in woodlands, so that by the start of the next season, our populations have been reduced, so we've got less pressure on our crops. So Adam is going to give us an update on where that's got to and how much new information has been found. And we'll just move on to Adam. Now, Adam, we can see your presentation. That's great. We can't see you. So if you want to start your video at this point, that would be very helpful.
Hi, Scott. It says that my video started.
Yeah, we can see you. We can see you.
See me now.
It's
okay,
perfect.
great.
We can see you, and we can see your presentation.
Brilliant. All right. Thanks very much for the introduction. So hello, everyone. My name's Adam Walker, and I'm a research assistant in the entomology team at NIAB EMR. Today, I'm going to give an update on reducing overwintering populations of SWD as a new control strategy. So currently, control against SWD targets the pest during the growing season, but there's potential to control the pest between growing seasons where populations are more vulnerable. Okay. Has that moved on for you, Scott?
Yep, that's perfect. Thank you.
Good. Okay. So since SWD was first recorded in the UK in 2012, we've been studying the phenology of the pest in and around soft fruit crops using traps. And what our monitoring traps tell us is SWD activity increases early in the season during warmer days in the woodlands, and then it increases in the neighbouring crops around April time as the SWD get attracted by fruit volatiles. Then the catches remain quite low during the growing season, both in the woodlands and the soft fruit crops, due to competition from the soft fruit crop volatiles. Then numbers increase again as you come into the autumn time, as the cultivated and wild fruit fades, and it peaks in woodlands around mid-November time before SWD activity decreases into the winter as it gets cooler, and that's in the woodlands. So using this information, our hypothesis is if we deploy precision monitoring in woodlands and wild refuges neighbouring soft fruit crops at the correct trapping density, before the autumn time, we can intercept the SWD before they go into their winter inactivity. Then if these traps remain in place through the winter and into springtime, they can intercept SWD as activity increases. And if they remain in place during the growing season, they'll intercept SWD, and so you'll get lower numbers of SWD migrating into the neighbouring soft fruit crops, and hopefully those numbers will be more manageable for the growers. So in October 2019, we set up a precision monitoring trial in 6 soft fruit crops in England. And each crop is a replicate, or each farm, I should say, is a replicate block consisting of 2 plots, a treated plot and a control plot, as seen in the diagram. The treated plot consists of a small isolated pocket of woodland, shown as the pink square, containing a grid of 64 precision monitoring traps spaced at 8-meter intervals next to a soft fruit crop. Then the control plot is another small isolated pocket of woodland, shown as the yellow square, next to a separate soft fruit crop. And the control plot woodland contains no precision monitoring traps. So to monitor SWD counts between treatment and control and compare those numbers, we've been using Riga traps, those yogurt pot traps shown on the right-hand side. And Riga traps have been deployed in each woodland and their respective neighbouring crops. And every time we visit, we filter these traps and we count the number of male SWD in them. Sorry, male and female SWD. This graph here shows the results of SWD catches at each trap position from October 2019 to August 2021. On the horizontal axis, you can see the different Riga trap positions, and then on the vertical axis, it's the mean SWD per Riga trap. And what you can see from the bars on the graph is in the treated woodland compared to the control, we've had fewer catches where there's precision monitoring compared to the control woodland without. And then in the respective crops, in the treated crop, we've got just over half the amount of SWD caught that compared to the control crop. This graph here breaks that information down into the series of assessments since October 19 to August 2021. And this is comparing catches in the respective woodlands. The vertical axis is the mean SWD per Riga trap on a log 10 scale, so that you can see the difference between the lower and higher end catches. The full black line are catches in the control woodland, and the dashed black line are catches in the treated woodland. And what you can see from the graph is that catches in the treated woodland are lower than the control woodland most assessments. This is similar information, but comparing control and treated crop. Again, the vertical axis is on a log 10 scale. And again, what you can see is the dashed black line, the treated crop catches are generally lower than the control crop catches most assessments. So to monitor SWD catches in precision monitoring traps, we've been using a transect of traps in the precision monitoring woodlands. And those are shown on the diagram as the blue fill circles. And what we do every visit is we empty these 8 traps per treated woodland onto a tray, and we count the number of male SWD on them. The top graph here shows the results from those catches in the treated woodlands from October 2019 to August 2021. And the vertical axis is mean male SWD per transect trap on a regular scale. And the bottom graph shows mean SWD catches per Riga trap. And the treated woodland is the dashed black line again. And what you can see, catches in the top graph follow a similar pattern to catches on the dashed black line on the bottom graph. So to monitor SWD egg laying and find out if precision monitoring can reduce down the egg laying potential of the SWD population, we've been deploying Sentinel fruit traps. And we've deployed these in each respective woodland and their respective neighbouring crops in the springtime. What these traps consist of is a red delta trap containing fruit to attract SWD females that are around to come in and lay their eggs. Then these traps are brought back to the lab at NIAB EMR and incubated for 2 weeks, and we count the number of SWD adults and other fruit fly adults that emerge from this fruit. Now, we did this assessment in 2020, and we used defrosted raspberries for that. But what we were finding is we were getting a lot of egg laying from other fruit fly species and a lot of larval competition, and so very few SWD emerging from that fruit. And so it was difficult to find an effect of the treatments. So what we did this spring is we used ripening strawberry in these traps instead, in the hope that they'd be a bit harder, they would be a bit more favourable to SWD egg laying and development in incubation. And this graph shows the results of this year's Sentinel fruit deployments. So again, on the horizontal axis, we've got the different Sentinel fruit trap positions. And then on the vertical axis, we've got mean fruit fly emergence from Sentinel fruit. Now I say fruit fly because we're counting SWD and other fruit fly species again. In the bars, the lighter grey bar is the adult SWD emergence. And then the darker grey bar is the other fruit fly species emergence from the same fruit. And again, you can see the same problem has occurred. And probably during incubation, as the fruit has deteriorated under the incubating conditions that we have them in, which favour SWD, it looks like we've got higher larval competition from other fruit fly species and then higher adult emergence from the same fruit and very low SWD emergence. We haven't statistically analysed this data yet, but it looks unlikely that we'll see an effect if there is one to be found. Now, we've also been running habitat assessments around precision monitoring traps, because what we found during the trial is some traps seem to catch more SWD than others. And it might be in knowing information from the surrounding habitat around those traps and the positions, etc., of those traps, we might be able to direct growers where best to put their precision monitoring traps to optimize SWD catches and therefore control. So what we've done is in a 4-meter radius around every trap in every treated woodland, we were assessing the habitat, scoring it according to how suitable it is for SWD and how abundant it is in that 4-meter radius. And then we've been correlating that back to the respective SWD catches within that trap. And what we were able to do with that data is create a predictive model showing habitat score and then the predicted male SWD counts that we would get according to a rising habitat score. And what we found in both our summer and autumn assessments is that as habitat score increases, we get an increase in predicted male SWD catches, which effectively means if there is more suitable and abundant vegetation around the trap, you'll get higher catches of male SWD in those traps. And we also analysed individual species, and we found in summer, as bramble coverage increased, we got a significant increase in male SWD catches. We've also looked at the trap position as well and tried to correlate that to see if there is any correlation between male SWD catches and the position of the precision monitoring trap in treated woodlands. And what we found is traps nearest the crop caught significantly more male SWD traps on the perimeter nearest the crop caught significantly more SWD than perimeter traps in general. So those are shown as the yellow traps on that diagram on the left, and traps in the center of the configuration. But there's probably more analysis we could do on this one. So to conclude, our Riga trap catches have shown fewer SWD in woodlands and crops where there's precision monitoring than without. And that's from November 2019, when precision monitoring traps were deployed, till August 2021. Our spring Sentinel fruit assessments are inconclusive again, due to few numbers of adult SWD emerging from the fruit and much higher numbers of other fruit fly species, which we think outcompeted them again in the fruit at the larval stage. We found a significant positive correlation between the habitat score and the male SWD caught in the traps. And in summer, as we wrote the vegetation species down, we found as bramble coverage increased, we got a significant increase in male SWD catches. And looking at trap position, we found highest male SWD catches in traps on the woodland perimeter closest to the soft fruit crop than other traps in our precision monitoring configuration. Okay, thanks everyone for listening. Are there any questions?
Thank you very much, Adam. Yeah, we have a couple of questions. First of all, how close does a woodland need to be to a neighbouring crop to become a source of SWD?
Well, it's a good question. The woodlands that we've been evaluating in this trial are right next to the neighbouring crop. So we haven't actually been looking at woodlands further afield, but it would be something that we could look into in the future.
Okay, comment from Michelle Fountain. Adam, your data suggests that this is a woodland edge species. It would be good to now look at the natural enemies that are abundant in woodland edges, so we can encourage these predators. It also highlights the importance of not allowing spray drift onto woodland edges. Thank you for that comment, Michelle. Another one question, what was the treated woodland treated with?
So when I say treated, I mean it had precision monitoring traps deployed in it.
Thank you for clarifying that. I suppose my final sort of concluding question would be, are we at a stage, this is quite exciting, this information, and it has shown that we can reduce overwintering SWD close to a new plantation, but the fact was SWD numbers do build up later in the season again. Is it something, if I were a grower, would you be advising me or suggesting that I try and do some of this in neighbouring woodland over the winter?
I think it can't harm you to do it if you have the resources to put into it, but I would stress that we haven't got all the data from this trial. It's ongoing. We're doing it through this winter. So it's probably advisable to wait and see what our final analysis comes up with, and then we can advise better, more conclusively. But in the meantime, I would definitely advise growers to continue monitoring SWD in and around their soft fruit crops.
Okay, I've only got time for one more question, the other one that's come in here. Comment, interesting to hear that the non-SWD fruit fly species created competition and reduced the number of emerging SWD. Have you stumbled upon a possible control strategy?
So I think one of our PhD students, Trisna, had worked on that and looking at other fruit fly species volatiles as maybe a deterrent or repellent for SWD. I can't go into too much detail on that, but there's she might be able to expand on that.
Okay, Trisna's listening in, so she can answer that. There are some other questions still coming in. I'm afraid we will have to move on. So I'll thank you, Adam, for a splendid presentation. Thank you for your help. Do hang around just in case we need you for another few minutes. But we will now move on to our next talk. Bethan Shaw is known to just about everybody here because you've all seen her making lots of presentations on her fantastic SWD research in recent years. She's got two presentations for us this morning. The first of these is on parasitoids, something that's been questioned for many years now. Have we got any reliable native parasitoids that will control SWD? And Bethan will give us an update on the latest findings. Beth.
Thanks, Scott. Can you hear me and see the screen?
Yeah, you're loud and clear and we can see your screen. Thank you.
Brilliant. Thanks a lot, Scott. Yeah, so I've been asked to give an overview of some of the information that we've gathered over the past 12 months from a desk-based review that has been funded by the AHDB to gather a little bit more information about the native parasitoid species of SWD in the UK. The focus of this review has been the 6 species that have been identified from the various survey work that's been carried out over the past 5 or 6 years. And these are the species that have been found to actively parasitize spotted wing Drosophila in the Sentinel traps that we've been deploying in various projects. So the review's been a fantastic way to try and gather some more information about those species and gain some more understanding about their biology and how effective they could be as future biocontrol options for this pest. So I'm going to start off with the pupil parasitoids. The technicians in my group will laugh at me because my pronunciation of the Latin names is terrible. I spent a long time on YouTube yesterday trying to learn the proper pronunciation. So Vindemini, this is the first pupil parasitoid that we identified from the first very survey that we did. And it's got a very broad host range of primarily dipteran species, so flies in general. This species actually had the most information available in the literature review, and that's primarily due to the fact that it's introduced in many countries to control Mediterranean fruit fly. So a lot of the basic understanding of its biology has been gathered and examined, so it can be used as a biocontrol agent. It's a hyperparasitoid, so it can use other parasitoids as hosts while they're within another insect host, which could have a detrimental effect on other species that it's trying to control. From the survey work that we've been doing, we found it in agricultural and semi-wild habitats from June through to October. And other research groups have found that this parasitoid can locate SWD pupa in dropped fruit on the ground. This could cause a little bit of an issue. I'm sure you're all aware that we really hammer home the importance of good hygiene practices when it comes to controlling SWD, but it's likely that that dropped fruit that's being collected to improve hygiene could be containing the pupil parasitoid within it. It's got a relatively low fecundity, but saying that, it has been the most frequently found species that we've recovered throughout all the surveys that we've done. And just as an example, in 2017, it accounted for 83% of the individuals that we recovered from the beta traps that we were working with. So Heterotoma, this is another pupil parasitoid and not only targets dipteran hosts, but also Hymenoptera as well. It thrives in relatively low temperatures, so it would make it a realistic option for use in early summer and late spring to control SWD. We found it in woodlands and hedgerow areas from May through to October, so quite a long period of activity, but the peak of this particular species was in July. The final two pupil parasitoids, just to give an overview on, are the Trichopria prima and Trichopria modesta. Again, both pupil parasitoids, but very little information was available on these two species. From the work that we did in the survey, this was the first record that I've been able to find of them actively using SWD in the field. There is a small amount of work that's been done on Trichopria modesta in lab-based experiments, but the literature is so scarce for these species that we've not actually been able to gain much of an understanding from them. We've identified two of these species, two in 2020 of Modesta and two in 2018 of Prima. So it would be beneficial to have a little bit more information on these two species, and I'm not going to discuss them again in the rest of the presentation just because there's so little to share. Moving on to the larval parasitoids, the Heterotoma has been the most abundant larval parasitoids that we've found throughout the surveys. It's a generalist parasitoid species of Drosophila, and it targets the very young larvae, so between up to 48 hours after egg hatch. So for SWD, that's the first and second instar. It's got a very strong attraction to SWD larvae, but actually results in very low survival of the parasitoid offspring. And this is due to a process called encapsulation, whereby the female parasitoid lays her eggs in the SWD larvae, and the immune response of the larvae actually enclose the egg with cells and basically suffocate the parasitoid egg and prevent the survival of the parasitoid. But in many cases, this does actually result in the death of SWD as well. So it's worth keeping in mind that as this is a generalist parasitoid species, it can actually develop quite successfully on other Drosophila species that don't have such a strong immune response. It's got very high fecundity, which again, if you take into account the low survival in SWD, is a very positive thing. It's relatively long-lived as well compared to a lot of the other species, so it can be present in an area for quite a long time. We've primarily recovered it in woodlands and on the edges of orchards, but only for a very short window between mid-August and the beginning of September, so it's only active for we've only been able to find it active for a relatively short period of time. The final species, Tibidea, another larval parasitoid, it's got an extremely broad host range, and that's due to the fact that it's associated with host species that are associated with fermenting fruit. So anywhere that you're likely to find rotting fruits. So not only commercial areas where you might get a bit of drop fruit, but also in woodlands and hedgerows where wild fruit's not going to be harvested. It's got a negative relationship between the number of eggs that the females lay and its lifespan. So quite often it will retain its eggs until there are a decent density of hosts available, because otherwise it's just shortening its own lifespan for no population gain. It can be a superparasitoid, which makes it a little bit unreliable because it means it will lay its eggs in hosts that have already been parasitized. So it's almost not making the most of the host numbers that are available. We've recovered it during June for our surveys, and we've had really low emergence of offspring of this particular parasitoid from the culture work that I'll present in a minute. On the screen at the moment is a comparison of the numbers of parasitoids that we've recovered from traps deployed in various habitats from the 2017 survey. And what you'll notice is that we recovered these 4 common species from the wild habitats. And this kind of goes back to one of the questions regarding the precision monitoring work about the woodland edges and woodland centres. We actually found the majority of the parasitoids in the woodland centres, although we were still picking them up around the edges of the crops, the edges of the woodlands and hedgerows as well. You'll also notice that we did find quite high numbers of Vindemini in the commercial cropping areas, which was really positive because there had been some concern that we just weren't going to be detecting them in the commercial crops that we were surveying. I've just included a very brief bit of information about the work we've done looking at the survival of these parasitoid species in artificial cultures in SWD in the lab. And this comes back to looking at how appropriate they could be for future biocontrol options. So what we did, we took the parasitoids that emerged from the field trials and introduced them to cages of SWD, and then looked at the parasitoid population growth over time. And what we basically want to see is a number above 1 for the number of offspring per adult to show that the populations are actually able to expand. So for these species, only the top pupil parasitoid, Vindemini, was able to grow its population on SWD as a host. It's worth keeping in mind, though, that you could use other Drosophila species as an alternative source for mass production of these particular parasitoids. But there is also the concern that comes with that, in that they may become accustomed to targeting those other species. So if you deployed them in the field, they might hunt down Melanogaster instead, because that's what they've become accustomed to. They found that for Heterotoma, the larval parasitoid, that's not been the case so far, but for the other species, they've not actually investigated that in too much detail yet. This final slide on the information about the species that we've recovered is just to give an overview of the life traits of each of the different species. I think it's really important to look at the period of time that it's active for in the field, because that would give you an indication of when you would want to be deploying these as a biocontrol agent. As I say, for the population growth in cultures, that's referring to population growth on SWD, but it seems that the Vindemini would be the most appropriate, followed by the Heterotoma, which is quite a long period of activity from May through to October. So there's a couple of options that could be combined for biocontrol of SWD with parasitoids. It might be a couple of pupil parasitoids and a couple of larval parasitoids combined to give a longer period of control. Some of the other key findings from the review was there's still so little known about the species that we've identified, and it would be really beneficial to have a better understanding of, particularly of the less common parasitoids like the Trichopria, Prima and Modesta, because they could be really beneficial, but we just don't have enough information about them. It's clear that parasitoids solely won't control SWD, but it is likely to contribute to reducing population sizes. And as Michelle touched on before, a lot of the literature indicates that growers should be preventing drift into semi-natural habitats, because a lot of the parasitoids have been found to be quite sensitive to chemical plant protection products. And I've just taken a snapshot of information which looks at the impact that some of the more commonly used pesticides for SWD, the known impacts that they have on some aphid parasitoids. I wasn't able to find any reference to the parasitoids we're interested in, but you can see it's quite a broad range of impacts that those products can have on the different species. And finally, the last thing from the review is that nectar resources for parasitoids have been found to really improve movement of them through the crop. So it's almost like making a corridor or a channel for them to follow. They're quite, they don't travel very far from the nectar sites, so it's quite useful to have this corridor to get them further into the crop. And there's a little bit of information there about how to, what to consider when deploying wildflower options. The review that the HDB funded will be released hopefully before Christmas. I have just got a few more edits to do, but that will have far more information than what I've been able to talk to you about today. So finally, just to thank the Worshipful Company and the HDB for funding a lot of the survey work and the review, and the Berry Gardens field staff that helps identifying and collecting these species. And also thanks to the guys at the Natural History Museum for their identification skills. Thank you.
Thank you very much, Beth. We've got floods of questions coming in. We don't have perhaps time to do them all justice now, but first of all, could introductions of parasitides in field crops be likely with any of these species? Is that a commercial possibility in future?
I think that's where it's going. One of the main reasons we looked at gaining more information about these species that we've identified in the UK is because they're native, there's not going to be the issues associated, like the registration issues associated with trying to introduce a non-native parasitoid. So we know that there are some species like Trichopria, Drosophila that are very effective at controlling SWD in parts of Europe, but we've not been able to identify in the UK. So we're trying to investigate how efficacious our native species would be for then possible development as a biocontrol strategy at a later date, just because there's not going to be the issues associated with the registration getting it approved as there would be for the non-native species.
Okay, another question, are the parasitoids also attracted to Sentinel traps?
That's how we did the survey work to begin with. We were baiting traps with larvae and pupae of SWD and then recovering them and looking at what species were emerging. So yes, they are.
Okay, thank you, Beth. I think we'll need to stop there. Can I just point out to everyone, if you're not already doing so, you can look in the chat box, and some of the other questions that I haven't posted, Beth, are being answered by Michelle Fountain and her entomology team. So that's very kind of you, Michelle and colleagues. And Beth, and perhaps you can have a look in the chat as well and add to any comments there. So we're very grateful to you for doing that. Bethan, we'll be seeing you shortly because you've got another stint to do later on. So thank you for now, Bethan. We'll move on, and I'm very excited by the next topic. Many people have talked about sterile insect technique in recent years as a means of controlling spotted-winged Drosophila. There's been a lot of talk about it. There's been lots of discussion, but perhaps nothing in practice. But we have a solution. We have Glenn Slade with us this morning from Big Sis. Glenn has set up his company, Big Sis, and he knows all about sterile insect technique from his previous experience working in science. I'm very pleased to say that Glenn has been doing some positive work in the field with sterile insect technique this last season, which has been very kindly funded by Berry Gardens and supported by the staff and the team at NIAB EMR. Good morning, Glenn. You're going to tell us a bit about what you've been finding in practice this year.
Yeah, thank you, Scott, for the introduction. I hope you can see my slides.
We can indeed, thank you.
Fantastic. Well, good morning, everybody. I'm Glenn Slade, founder and CEO of Big Sis. We're a 4-year-old UK startup. And yes, we're all about the sterile insect technique. So today I'm going to tell you a bit about SIT and the field trial we've done this year to control SWD. Before I do that, let me repeat the credit Scott gave to Berry Gardens, who helpfully sponsored this work and also their growers provided the field, and particularly to Michelle Fountain and Adam Walker at NIAB EMR for their support during the project. So for anybody who's not familiar with SIT, come on, next slide, there we go. It works very simply. Basically, we produce lots of sterile male insects of the pest in question, SWD in this case. We release them into the field, which we wish to protect. They mate with the wild females in the area, and they then have no offspring, which prevents the explosion of the population that we normally see with pests. It sounds a bit fanciful, but it's been used for over 60 years. It's been used against dozens of pests from different insect families. So it's very much a general method. And the reason it can be so successful is that it's harnessing the mating instinct of the male insects that we release. They go and find those wild females in the natural habitat. And so they can be very effective, bit of a guided missile. There's two big reasons why I think SIT can make a big contribution to agriculture. The first one is there's not a lot of biocontrols that you can honestly say are more effective than chemicals, and in turn can achieve an improved crop yield through the season. And I've tried to show in this graphic the reason for that. So the green parts, the SIT, is applied frequently throughout the season. And you can see the green line shows that the pest infestation stays low. So in other words, used preventatively, as each wild female is emerging, they're much more likely to meet a sterile insect and therefore not have offspring than a wild one, which would allow the population explosion. And that's a contrast with traditional chemical control, where actually you wait for the pest to build up before it becomes economical to make an insecticide spray and bring the pest under control again. And the area under that orange curve is actually yield loss. And so we may have become accustomed over the years to calling a few % of yield loss as good control, but actually SIT has been shown to achieve much better control than that. I've given an example data here from the Codling Moth project that's been running for a long time in Canada. So better yield and less insecticide use. And the other big reason why I like SIT is its sustainability. So sterile male insects are species specific and non-toxic. So they're excellent for biodiversity, as well as reducing the use of chemicals and increasing yield, so there's less food waste. On top of that, Big Sis is using local native strains. So to be absolutely clear, we are not a GMO solution. We are taking English SWD for England, Californian SWD for California. And what that means is in a number of areas, the regulators actually require no permitting at all for trials or commercial sales. SIT has got a very long safety track record. And because we're using local strains, we're not risking introducing either insecticide resistance or viruses or other pathogens. So since we're approved for sale, the main thing we need to do is to scale up production so that we can do a commercial launch. So we've been working on this for a number of years already, and we are currently building our first automated SWD sterile male production plant, which will be used to run large field trials next summer. And we're aiming for a full commercial launch for the season of 2023. What we've been able to do this season is to use a laborious manual method to produce enough sterile male SWD to conduct a field trial on a 6-hectare site in Kent, which has ever-bearing strawberries. And you can see in this little map the red dots, which are the where we located red sticky traps with a dry lure, which was our principal trapping method. And we put in a number of more traditional Droso traps, bucket traps, as a secondary metric. And what we also had, of course, not shown on the chart, are 2 plots that were not treated with sterile insect technique. They were 1 hectare each. One was a few hundred meters away, the other was over 1 kilometer away. And we were doing releases of our sterile insects from about mid-April, so definitely making sure we got in there early, and we continued through to mid-October when harvest ended. And I'm very pleased to say that we achieved our goal, which was season-long control of SWD. So the green treated line at the bottom is showing the SWD prevalence on our treated site as measured by the average number of female SWD per sticky trap. And it's showing a 3-week moving average on these lines. The suppression was up to 91% compared to the sites that did not receive sterile insect technique. And we're showing that data in mid-August because just after that, there was an insecticide spray against thrips on this treated site. And just for reference, the orange line, the other site, also had over 70% suppression. Those lines can be a bit deceptive in terms of the maths. And I think we would have got a higher result, except for the timing of these sprays. But you'll also see on this chart that when the control sites were sprayed just after mid-September, they did not, even after the spray, achieve an SWD level anywhere near what we achieved on the treated site all season. So we're very excited by that result. To share a few of our secrets of success, I've already mentioned that prevention is better than cure. We got in there early and made sure there were never enough SWD to really take off. So throughout the season, the number of sterile males we were releasing could always compete with the wild males for the females that were present. The second thing we did was we used these heat maps you can see on the right at least once a week so that we could target where we were releasing our sterile males. So the top 2 graphics are the untreated control sites, and therefore sort of showing red because there's a lot of SWD there. The bottom graphic is the treated area. And basically what we'd be doing is releasing sterile males throughout those tunnels, but releasing relatively more where that warm yellow spot is, and also on the bluey-greeny areas compared to the average of the site. So that made better use of our resources. And I haven't shown on these charts the boundary, but that would of course have a very high infestation of SWD as the season goes on. But because we were able to treat that boundary as well, again, unlike many other controls, we were able to keep that SWD population low in the boundary, and that reduced the invasion pressure on the crop tunnels. A couple of other quick advantages, more from the grower perspective. Firstly, that Big Sis does all the weekly releases, not just in the trials here, but that will also be our commercial model. By default, we will do that. So there is no extra resource requirement. I'm picking up on one of the points made in the first presentation. And secondly, that SIT is compatible with all other controls. So even other SWD controls are welcome to be used alongside. If an insecticide spray takes out some of our sterile males, we'll be releasing some more the next week, and they'll take out the wild ones too. So finally, what we'll be doing next then is continuing this work going into next season. I'm pleased to say that, again, in conjunction with Berry Gardens and NIAB EMR, we've received some funding from Innovate UK to do much more extensive development work on our SIT solution. We've started that already, and during the winter months, we'll be working on mating competitiveness and looking at the different factors that can affect the fitness of our flies before they're released, for example, how old they are, how sterile they are, and so on. And then particularly working towards 3 commercial trials during the summer, so potentially on cherries and raspberries, as well as repeating the strawberry plot that we worked on this year. In addition to that, we'll be doing some field cage studies to study the dynamics of our flies in more detail. And picking up on this point about all year control, we'll be looking at the interaction of SIT and the winter morph SWD in the 22-23 winter season. If we've got enough production, we will be able to do a few small commercial plots as well, if growers are interested. But I will leave it there and leave a bit of time for questions. So thank you very much.
Glenn, thank you so much indeed. Very exciting developments there. I'm sure everybody will agree. Just a quick question from me. You kindly set out the timescale at the beginning as to when this might become a commercial reality, and that might be 2 years away, perhaps. Would you, as your organisation, supply the service so you would continue to supply the sterile males for growers to release?
That's right, Scott. We'll be providing not just the sterile males as insects, we will actually do the releases on the farm. It's right. So it's a sort of complete service. And so really we would just need to agree with the grower, which day of the week or half day of the week we'll be on the field, so that there wouldn't be, for example, other fungicide sprays going on, so that we wouldn't be able to be in the tunnels. And then we would, deliver that complete service and keep them posted on the infestation levels as the season progresses.
That's helpful, thank you. Another couple of questions. Glenn, do sterile males live as long as non-sterile males?
They will not live quite as long in the wild because they are lab adapted, they're lab reared or factory reared, but they live 3 to 7 days, and we adjust our release numbers according to the longevity we're seeing in the field. Okay,
and an obvious question, how do you sterilize the males?
Using X-rays.
Okay, and final question here, what is the life cycle for SWD males and timing of introduction? I'm not sure what that last comment means, but perhaps you can respond to that.
So SWD, we are introducing right at the start of the season because we want to use this preventatively. So we started in mid-April this year. That may have been even too early, especially in a cold winter. But we want to start releasing the sterile males before the first wild females are emerging. And then we are releasing throughout the season, right until after harvest. So we are looking to maintain effectively a standing population of sterile males, which we repeatedly top up every week. And then that will compete with the sterile males on their life cycle, and we monitor the ratio of sterile to wild throughout the season. I hope that answers the question.
Yeah, I hope so. If you do look in the chat box, there is another second part to that question, which you might understand better than me. So you might be willing to respond in the chat. But Glenn, for now, thank you so much for sharing that really exciting information with us. I'm very grateful to you. Things are coming together here, because Adam has found a way of reducing overwintering populations of SWD. Bethan is finding ways that we can try and encourage parasitoids. And Glenn is now finding a new technique to try and reduce populations throughout the season. Leads us nicely on to our next speaker, Trisna Tungadi, who is possibly new to many of you. She works at NIAB EMR. She's a postdoctorate researcher, and she's been looking at egg-laying deterrents. What deters Drosophila suzukii from laying its eggs? Because if we could find out how we can deter this, then again, it's another tool in the box. Good morning, Trisna. It's all yours.
Yeah, thank you, Scott. Hi, morning, everyone. So I'm Trishna, and I'm a postdoc working in the entomology group on a BBSRC funded project. So as Scott mentioned, I'm going to talk about what deters SWD from laying eggs. because we know that the main problem SWD causes when they lay eggs and then the eggs hatch into larvae. So going back a bit to Adam's earlier presentation, where he mentioned that non-SWD fruit fly species can create competition and reduce the number of SWD caught in the trap. And Gloria also posted a question in the chat box earlier asking that SWD doesn't seem to like habitat sharing with other species. So yes, that's what I'm looking at for this project. So SWD do normally lay eggs on fruits that are still on the trees or ripening fruit. However, at the end of the growing season or when there are less fruits that are still on the trees, SWD can and they will lay eggs on off-ripe and rotten fruits on the ground. And this is when SWD will encounter other non-SWD Drosophila species. For example, Drosophila melanogaster are usually utilizing these rotten fruits. So the aim and the hypothesis of the project is we want to investigate how interaction with other non-SWD Drosophila species will affect, in particular, SWD egg laying. And what can we learn from this that perhaps we can take further to optimize current control methods? So the work I'm doing is, and I'm going to present, is all done in the lab. So we do choice tests where SWD is offered a choice to lay eggs on 2 different petri dishes that are filled with fly food. And this is the same fly food that I use to keep the Drosophila cultures in the lab. So one of the plate has been exposed to Drosophila melanogaster for 2 days. So during these 2 days, Drosophila melanogaster is free to feed on the plate. They can lay eggs, and some of those eggs would then hatch into larvae. So after 2 days, melanogaster is removed, and then I put this plate into a plastic box, and then next to it, I place another plate that has never been exposed to melanogaster before. So the next step of the experiment is I then introduce SWD into this box, and then I leave SWD there for 2 days. And during that time, SWD has the option whether they want to lay eggs on one plate or on the other. After 2 days, I remove all SWD and I simply count the number of eggs present on both plates. So what we found is that, as you can see here, SWD clearly laid fewer eggs on plates that have been visited and exposed to melanogaster previously. And the blank media is the plate that has never been visited by melanogaster, and SWD lay more eggs on that plate. So this particular finding was initially found by Beth and Shaw a few years back, and then I took on the project to investigate further. So what we thought from this is that melanogaster have left something on the plate, a compound that is detected by SWD, which then deter them from laying eggs. We wanted to know what the compound is and where the compounds come from melanogaster. So the project started by me performing multiple choice tests by simply offering the plate to either melanogaster male only, female only, eggs only, or larvae only, because we wanted to know which life stage of melanogaster is the source of this egg-laying deterrent for SWD. And by doing this multiple choice tests, I found that the most consistent result and the one that always leads to fewer eggs being laid by SWD is the presence of larvae of melanogaster on the plate. And here, as you can see on the left-hand side, so when only live melanogaster larvae are present on the plate, SWD consistently laid fewer eggs on those plates compared to plates that have that doesn't contain any larvae. And interestingly, when I only placed dead melanogaster larvae that was killed beforehand and offered them to SWD, SWD doesn't seem to be deterred to lay eggs on those plates. So what we can conclude from this finding is that there's something that live melanogaster larvae either left on the plate or whether it comes from live Melanogaster larvae itself that is detected by SWD that cause them to lay less eggs. So the next stage of the experiment is trying to find out whether cuticular hydrocarbons that are present on the bodies of the flies or larvae can be the source of this egg-laying deterrent signal. So cuticular hydrocarbons are compounds that are present naturally on the bodies of all insects. It protects them from dying out, and it also acts as communication signal between insects. So in this part of the project, I simply extracted and analyzed and identified different cuticular hydrocarbons present on Melanogaster or SWD. And the work is done with our collaborators at NRI in Greenwich. As you can see on the traces, so the traces showing different chemical compounds and different quantity. So we know that there are different quantity and different compounds present on both Melanogaster and SWD male and female. And I've taken the work further by testing whether applying this chemical compounds onto the plate, whether it will deter SWD from laying eggs or not. And the answer is no. So it doesn't seem to be that cuticular hydrocarbons present from the bodies of the flies are the one that is causing the egg-laying deterrent effect to SWD. So in summary, we know that it appears that presence of live larvae of Melanogaster deter SWD from laying eggs. It doesn't seem to be coming from the bodies of the larvae or the flies. And currently, I'm just investigating whether it's something that the larvae secrete on the media when they're feeding, perhaps, that cause the egg-laying deterrent effect. And also, I'm testing other non-SWD Drosophila species beside Melanogaster to see whether we are seeing the same effect or not. And I think that's it from me. Thank you, everyone.
Thank you, Trisna. Just a quick question from me is how much longer do you have to do this work, or is there an end point in sight?
So it's a 3-year project, and currently about 7 or 8 months left. So
okay. So some exciting results, but a slight disappointment in the middle. But there is time to turn it into a positive, hopefully, and maybe you'll be able to share the remaining 7 or 8 months with us this time next year.
Yeah, sounds good. Thank you, Scott.
Wonderful. Okay, let's move on. Time is short, and we're back to Bethan now for our final talk on SWD and our final talk of this session. Biotremology is something that many of you may not have heard of. It's something that Bethan introduced to me and my colleagues at AHDB, and we felt quite excited about this, and we felt it was worth funding. So AHDB and also the Worshipful Company of Fruiterers combined to fund this bit of work, and Bethan will explain more about biotremology and how it might be applied for SWD. Beth.
Thanks, Scott. Yes, so we had 2 contributions, one from the AHDB and one from the Worshipful Company of Fruiterers, to fund both lab and field work to explore this concept of biotremology. And the theory behind biotremology is that some insects use vibratory signals in communication and courtship. And some groups have found that if people can disrupt these vibratory signals, they can then disrupt the impact of these insects in crops, particularly as pest insects. So we had a couple of aims within our project. First of all was to try and develop a piece of kit that could actually employ the concept of biotremology, which could be used in both a lab and in a field setting. So we had kind of a technical element in actually building this piece of kit. And then that was taken further into looking at testing the efficacy of this kit to disrupt mating success in Lagusrugal apennis, the European tarnished plant bug, and also oviposition behaviour in Drosophila suzukii in strawberry. So to start with, we had to think about how we were going to actually develop this piece of kit, and vibrations can be made by many different methods, and we felt that our 2 options for trying to use this in a cropping situation were going to be through either sound or movement. So for sound, if you've got a really bassy speaker, you can quite often feel the vibrations through the floor. But for our particular project, we decided to go for the use of movement instead. And the image you can see at the moment is a small motor that's got a weighted cam attached to it. And it's basically a small version of what you have in your mobile phone that causes the vibrations when someone rings. So the weighted cam spins around and it produces this movement. So to start in the lab, we developed a tabletop system that we could attach these motors to, which would then send the vibration signals through whatever we put onto this particular table. And it was a Perspex sheet supported by quite large springs, actually. So the idea is that we could put samples with SWD or Lagusrugal penis into boxes within this table, and we could send the same signals throughout the substrate that was being placed on there. So this is just an overview of the setup that we had in the lab. I think you can see my mouse, but we have 3 motors attached to the table, which are bulldog clipped down. And because we knew that eventually we wanted to move this device into the field, we started by trying to use battery-powered solar packs so that we didn't have to rely on mains energy when we got into using this kit in a more remote setting. So as I say, this is the table that's treated with biotremology. We've then got a control tray next to it in which no signals were being sent through. And on both of these trays, you can see these small Perspex boxes, and they hold the cornmeal media that Trishna was just talking about that we use for oviposition for SWD. So each of those 12 boxes has got a small Petri dish with this media in, and we have cohorts of flies that are introduced into each of the boxes. And the idea was we wanted to see if we could reduce the number of eggs that were being laid in that media on the treated table compared to those that were on the control table. And I've just got a very short video, which I really hope works, just to show the kit in action. So you can see the motor whirring on the left-hand side of the screen. I'm not sure if you can hear the sound, but it is also producing a small amount of sound. What you'll notice as we pan over to the control tray is this Petri dish that's not within a box with a small disc on it. And that's actually a receptor that is measuring vibration frequency. So we were using that to make sure that we weren't accidentally causing the control tray to send out a vibratory signal as well. And what we could do is we could use that receptor to take different vibration readings wherever we placed it, basically. And we then collated that information on the computer. We were also able to use this receptor on the plants themselves to check that the table was actually sending the signals through the substrates that we're working on. In this instant, we've got the receptor attached to a leaf, so it's just held on there with a paper clip. The motor's whirring, and what you can see on the screen is what the computer logs. So there is a motor controller, and we can increase and decrease the frequency. You can't see it very clearly on the screen, but along the bottom, that's the hertz of the signal that's being sent through. So we're able to detect on the plants we're able to detect on different parts of the plant what signal is actually getting through to the extremities. Because it might be that you're able to really well detect the signal in the substrate, but it's not actually reaching the fruit or the leaves that we're trying to get the signal through to. So as I said, in the lab, we first looked at the impact that this treatment had on the number of SWD eggs that were being laid. And what we found in 6 of the experiments that we did, we found a reduction in the number of eggs that were laid in 4 out of the 6 experiments. We actually found an increase in 1 of the 6 experiments and no significant difference in another. And we were really enthusiastic about this result because we've gone from having basically a glorified Blue Peter project into being able to reduce the number of eggs that are being laid by SWD in this lab setting. It's worth saying that in the lab setting, it was a no choice experiment. So the females either had to lay eggs or not lay eggs. It's not like they had a choice between a treated and an untreated area. So as we took this into the field, we actually started to explore the choice element of using biotremology. Moving it into the field came with its own issues. As I say, we didn't have mains power, so we had to work out how to actually drive the motors in the field. And we settled on using these leisure batteries that are used for camper vans. And we were able to get about a minimum of 5 days worth of charge from these batteries, and they were charging 12 motors that were attached to the tape system supporting these tabletop strawberries. I think you can see in the bottom right image. We cable tied them to the tapes, and that's how we were sending the vibratory signal through. We had these custom built tabletop strawberry sections made within a enclosed insect proof tunnel. And we had this as a choice element. So one side of the tunnel, I think you can see my mouse in the top right hand side, one side of the tunnel was treated with the biotromology kit, and the other side was just the same tabletop strawberry system set up, but with no signals being sent through. And because we could only power these motors for 5 days at a time, we bagged all the fruit when the motors weren't running to prevent any damage being caused while we weren't running the experiment. We made sure that we were still able to detect the biotromology frequencies on different parts of the plant using those receptors again. And luckily, when we looked over at the control tunnel, the control table, we weren't detecting any of the frequencies. So the green line on this bottom figure is the frequency being detected on strawberry fruit that were in contact with the support tape. But luckily we weren't accidentally, causing vibration signals to be detected on the control tables. We then released groups of SWD and Lagusrugal apenis into these tunnels and looked at the amount of damage that we were detecting on the fruit over a period of several weeks. So I'm just going to show you the results from the Lagusrugal apenis assessment. And for that, we were looking at the amount of fruit that had cat-facing damage. And what we wanted to find was an increase in the amount of fruit that showed no damage in the treated areas compared to that on the control areas. So we're looking for this, this grade 1 capsid damage scale. And what we found is we were able to get an increase in the amount of undamaged fruit in the treated areas in 2 of the assessments. We didn't find much difference in the other assessments, but I think, although we tried to account for this, I think that might be due to pollination more than capsid damage itself. We did have beehives in the tunnel, and we did do some hand pollination as well, but I don't think the hives were quite as effective as they could have been. But we were still really, really enthusiastic to find that we got an increase in undamaged fruit where we were using this biotromology kit. So kind of the conclusions and the next steps from the work is we need to be able to optimize this method to use vibratory signals to disrupt pests. It might be that we need to alter the transmissions of the vibrations, and we know that for particularly like things that are grown on post and wire systems, sending the vibratory signal through the wire supports would be a fantastic way of getting the signal through a larger area of crop. The car battery system worked great for our small trial, but it's not realistic for a grower setup. So it might be that it needs to be combined with solar to power the kit. As I said, we'd like to test this in other crops on different growing systems like the post and wire to see how that either improves or enhances the efficacy. We don't know if it's density dependent on the pests. So biotromology might work while there's low or minimal pest pressure, but there's no indication of how it would be affected if you have very high pest pressure on crops. There's also other species that we could investigate and see how this is impacting them. And I think that follows on to saying that we're not sure how this is going to impact beneficial insects. So far, I've not found any reference to how this could deter or impact pollinators, for example, or other predatory insects that are being introduced in crops. So that is more work that needs to be investigated. But this funding from the Worshipful Company and AHDB has been a really great start to ticking off some of the fundamental questions that are associated with the use of biotromology. So finally, I just need to thank Graham Caspel and the farm team who built the tabletop system for us, and the technical staff that have done this trial on my behalf, particularly Jonah, Ben and Francesca. They have been wonderful, and Adrian for help on fixing the motors, because they broke quite a lot during our time. But thank you ever so much. And if there are any questions, I think I've gone a little bit over, but you can always send them to me directly.
No problem, Beth. I think there's a couple of questions which you can have a look at in the chat, although I think you've actually answered them to some extent, but perhaps you can have a look at those. I just reiterate what Beth says. This was a very small project, very small amount of funding. I'm still excited about this, and I do think that we need to find a way to continue this investigation, because I think there's a lot riding on this, and it could be very positive. Thank you, Beth. I just want to finish this session by saying that all this research is fantastic, and hopefully it will lead us to improved ways of controlling spotted wing Drosophila. My crop protection colleague in AHDB, Adam Doxford, is hugely dependent on this ongoing research, because without it, CRD and the Expert Committee on Pesticides are never prepared to issue us with continuing emergency authorizations for crop protection products for spotted wing Drosophila. So not only are we obviously trying to find an alternative control method for SWD, alternative to traditional crop protection products, but the ongoing work is absolutely essential to support our applications or submissions for emergency authorizations. So it's all good. It's all good progress that we're making in every respect. Thank you, Beth. Thank you.